so nice. I've been to all four of these, and I think they've been fantastic meetings. So Thomas charged me with giving the same talk I gave last year, and I hate giving the same talk. So I've changed it up a bit. I've got some slides from last year, but I've tried to make it a little bit more unique for this year. So these are my disclosures. The problem with ITP, the central problem with ITP is a clinical problem, of course. The diagnosis of ITP is a huge problem. We have no biomarkers for this disorder. You only get an isolated platelet count. There's no laboratory test for this disease, and that makes the pathophysiology and the clinical aspects of this disease very difficult because if you diagnose with ITP, within a year, 20 to 25% of your patients are going to come down with another immune disorder. So they're secondary ITPs. And this really complicates the problem with trying to understand primary ITP versus secondary. And of course, the diagnosis, the phases of diagnosis are here on the left, and they're new from the old categories, newly diagnosed, persistent, and chronic. And on the right is from Doug Seen's blood paper almost 10 years ago now, or 15 years ago, showing that in fact, maybe 70 to 80% of the patients you diagnose are really primary ITP. The rest of them have other immune disorders, and that's the problem. They complicate not only therapeutic responses, but also trying to understand the pathophysiology of primary ITP. So these issues that I'm talking about clinically, and this is the only real clinical slide I'm going to show, probably relate to treatment failures or refractoriness in this disorder. So animal models, their relevance to these diagnostic issues. So the first thing we all know about animals is that they don't look like humans at all. These are humans on the left, as you can see, and these are mice on the right. And you can see that, in fact, they don't look the same at all. However, they share a very common immune system. Most of what we know about human immunity has come from studying animals like mice, and they have all of the characteristics in the immune system that are very similar. There are, of course, slight differences between them, but we can understand a lot about human immunology by studying animals like mice, particularly mice, because they're so well characterized. In veterinary medicine, ITP is also very prevalent. It's not just a disorder of humans. There are thousands of clinical reports on ITP in domesticated animals, from dogs, cats, all the way to horses. We treat veterinary ITP the same as human ITP. They start off with corticosteroids. They could be given IVIG, and we certainly treat them in a very similar manner. But it's not unique to humans, so animals do get it. The interesting thing is that laboratory rodents, we haven't been able to find ITP in laboratory rodents. We just don't spontaneously get it in mouse colonies. It may be there, but we don't see it because mice are quite resistant to low platelet counts, and they don't tend to bleed when they have low platelet counts. I think it might be due to the breeding schemes between domesticated animals versus laboratory animals. Domesticated animals are line bred, whereas laboratory animals are inbred by brother-sister mating. So that may be the reason why we don't see ITP. So we have to manipulate the animals in order to make them thrombocytopenic. And I'm just going to go through a couple of models about how that works. Is it valid to study ITP in animals? Well, on the left is the famous or infamous Harrington experiment, where in the 1951 they infused each other, medical students and residents who probably volunteered, I would imagine, to get plasma from patients with ITP. And you can see they all had very precipitous drops in their platelet counts on the left. Mice, if you give them an antiplatelet antibody analogous to a plasma infusion, they get this same smile. They get thrombocytopenic within 24 hours of the infusion, and then the platelet counts rescue. So they do similar things when we give antiplatelet antibodies to these animals. Now, there's four major categories of of ITP animal models. The passive transfer model is probably the oldest, and it's still used today, and that's where you inject an antiplatelet antibody into the animal. They become thrombocytopenic, and you can certainly test therapeutic responses to that antiplatelet antibody. The active immune model I'm going to talk more about in this talk. This is where we can take knockout mice of particular platelet antigens, immunize them against that platelet antigen, and then transfer their spleen cells into immunocompromised animals like skid mice, and I'll go through that in a moment. There are secondary autoimmune ITP models. The most famous is the NZW by BSXB mouse, where it's a lupus background, but yet they get thrombocytopenic and they have an autoimmune-like platelet disorder. There's drug-induced type models, and there's infectious ITP models. These acute models like LDH virus infection causes thrombocytopenia in mice. So there's a number of different models that we can study in animals, but I'm going to really stick to the first two and just briefly describe them. So the passive model is incredibly simple. You take a mouse and you give it an antiplatelet antibody. The most canonical platelet antibody that we use in the literature is this clone called mReg30. And when we inject it within 24 hours, we can bleed the animal. And you can see generally how the platelet counts look on the right. This is the animal becomes thrombocytopenic within a day. And then if you don't give them any treatment, their platelet counts will rescue within 2 to 3 days. If you give these animals IVIG, you can completely suppress that thrombocytopenia, so they rescue the platelet count. So this type of model is amenable to certain therapies that we can do to look at how we can combat antiplatelet antibody-induced thrombocytopenia. The active model, my lab developed it about 15 years ago, and it's a little bit more complicated, but it's not that difficult to understand. We take a mouse that's knocked out of a particular platelet protein. The first one was CD61, that's the 3A chain of glycoprotein 2B3A. And we immunize that animal with wild-type platelets that contain that antigen. And so they become very strongly immune. Some of their titers against anti-glycoprotein 3A can reach up to 100,000 in titer. And once we find that they're immune, we sacrifice the animals, we remove their spleen, and we can take their spleens and transfer them into skid mice that have been sort of manipulated a little bit. They get a little bit of a radiation in the first original model, and they get an anti-NK cell antibody to get rid of their natural killer cells. And then all we do is we monitor these animals over 4 weeks. We bleed them every week, and we monitor their platelet counts. And we can do all sorts of other things that I'll talk about. We can manipulate the spleen cells that we put into these animals. We can remove things. We can remove B cells from these spleen cells and put them in. We can remove T cells from the spleens and put them in. And that really affects the way the skid mice are going to get ITP. And these mice, if we want, we can make them bleed profusely. The control mouse is up on the top left, and you can see the brain, for example, in this case is very nice and pink and clean. But the ITP mouse, scruffled hair, piloerection is taking place. They're bleeding everywhere, and this is a large dose of spleen cells we've given these animals. They have petechiae, they have subcutaneous bleeding. Their spleen is enlarged, which is not what happens in human ITP, but that's just a characteristic of giving immune cells to an immunodeficient mouse. It has nothing to do with the ITP itself. So if we lower the dose of spleen cells, we can make the ITP much milder in these animals, similar to what it would be like in humans. This is the typical platelet count that we originally described in these animals. In the top left, when we just put non-depleted spleen cells in, the animals become thrombocytopenic in the first week, and they remain thrombocytopenic. And this was a large dose of spleen cells, 50,000 spleen cells given. And in fact, we couldn't get any more platelet counts because most of the mice died of bleeding. If we deplete CD4 positive helper cells, we completely abrogate the response. They do become thrombocytopenic in the first week, but that's due to the irradiation that we give the animals. But they rescue their platelet counts, and this is the typical control response that we see. So the thrombocytopenia is CD4 dependent. That's the first thing that we noticed. If we remove CD8 positive T cells from the spleen cells before we give them, this is what we call antibody-mediated ITP, and they get a very similar thrombocytopenic pattern to the non-depleted cells. They bleed profusely, and they bleed and they're thrombocytopenic till about day 14. Interestingly, we think it's an antibody-mediated disease, but in fact, if you get rid of the B cells of the spleen and transfer them, the animals still become thrombocytopenic, and this is what we call CTL-mediated thrombocytopenia. And this is the real advantage to this animal model, because it not only shows antibody-mediated ITP, but it shows T-cell-mediated ITP. So we can actually look at therapies against B cells and T cells in these animals. And what's interesting, the original description was in fact this, I'm not going to show it to you, but the antibody-mediated ITP was completely sensitive to IVIG therapy. It rescued the platelet counts. The mice remained healthy even though they got a large dose of these spleen cells. The CTL-mediated was not responsive to IVIG at all. They still remained thrombocytopenic and their platelet counts never recovered. So that first maybe suggested to us, is this why some patients fail therapy? Because the mechanisms of thrombocytopenia may be different in some of these patients, T-cell versus antibody-mediated. And we're still trying to understand that, whether or not that's true or not. The bone marrow, like in humans, in these mice, is completely abnormal. Down on the bottom left is a large dose. You can see that there's a megakaryocytopenia. There's no megakaryocytes in the bone marrow. They've all been destroyed. On the right is a lower dose, 15,000 cells. And you can now see that there's megakaryocytes there, but they're completely abnormal. And in fact, they're expressing apoptotic markers, just like in human ITP. So now we have an animal model that we can not only study different mechanisms of thrombocytopenia, but we can look at the bone marrow of these animals and determine, how these immune responses affect the bone marrow. So the similarities of this animal model to humans are quite profound, I think. First off, they show B cell and T cell mediated thrombocytopenia, like I showed you. They have bone marrow megakaryocyte abnormalities. They bleed, and it can be anywhere from mild to severe, depending on the dose of spleen cells that we give to the SCID mouse. And like in humans, they have peripheral Treg deficiency and MSDC, myeloid-derived suppressor cell deficiencies, the same that happens in humans. And this deficiency that we see in the SCID mice isn't due to a reduction in these cell numbers. It's just that they've been sequestered to the spleen and in some cases, or to the thymus and in some cases into the bone marrow. Therapy responses in these animals, we've done a lot of work on different therapies, the same therapies that we do in humans. They respond to steroids. IVIG, antibody-mediated thrombocytopenia, responds, but T cells doesn't. They respond to Rituxan. They respond to platelet transfusion, freeze-dried platelets we've looked at. They also respond very effectively to TPORAs. So this animal model, and I know this for sure, is an excellent preclinical therapy model because I've had several pharmaceutical companies contact me and they want to test their new therapies in this animal model. And we've done, for example, we finished up the VA736, the ianalumab antibody, and in fact, they respond to this antibody quite nicely. So this is what we're doing with this animal model now, trying to test new therapies to see whether or not they respond. So it's a great preclinical model for therapeutic responses in ITP. Can we use this animal model to study refractoriness? Refractoriness is the big question in ITP. Why do patients become refractory? What's the mechanism of refractoriness? And in fact, we did a systematic literature review a few years ago. This is, of course, the pathophysiology of ITP, where somehow there's a presentation event of platelet autoantigens to CD4 positive helper cells. This drives the B cells to differentiate into plasma cells to secrete autoantibodies, and these autoantibodies bind to platelets and they can be destroyed by the macrophages or by complement. And in fact, they also generate CTLs, and these CTLs are very good at migrating into the bone marrow and destroying the megakaryocytes. And these are all, this is certainly not a complete list. Drew and I drew this up. We can probably put more therapies in here now, but it gives you an idea of the different therapies that are used in human and where they basically work, we think, in the pathophysiological response. And many of the therapies that pharmaceutical industries are now looking at are based on this pathophysiology that you see here. So I think, we've learned a lot from mouse models on this pathophysiology. Now, I said we did Karina Yazdan-Bakish and myself did a systematic review. And in fact, when you look at the refractory literature in ITP, both at the human and at the mouse level, it seems that patients with ITP that are refractory have really significant abnormalities both in T cells and in myeloid-derived suppressor cells. So these cells appear to be at the center of refractoriness. And there's a whole variety of characteristics of these cells that we've looked at. And so the question is, does this active model, can we sort of test some of these therapies with regards to refractoriness in ITP? The first model was in fact by Hey-Yun's group, showing that in fact that CD8 T cells seem to be required for an efficacious response to steroid therapy. And this is a rather complicated graph, but in fact, if you deplete the mice of the spleen cells of CD8s and put them into the SCID mice, they don't respond to dexamethasone very well at all, not as well as if you don't deplete. And in fact, so the question then becomes, how do these CD8 cells, or how are they required for responses to a steroid like dexamethasone? And they're actively doing work in this model to try and understand that. So that's one way that we can look at refractoriness in this model. The other one is just been published a few years ago from Ming Hu's group. And it seems that in these MDSC populations, there's an impaired aerobic metabolism. And it seems to participate in the pathogenesis of, again, glucocorticoid resistance. And you can see that in mice that, in fact, are given an inhibitor of the glucocorticoid receptor, they don't respond at all to, in this case, it's dexamethasone again. But the wild type mice that are ITP, they do respond to the steroid. And this is all the different subsets that seem to change in number based on whether or not you inhibit the glucocorticoid receptor. So we can really get into a protein level and a molecular level in these animals to try and tease out various mechanisms of potential refractoriness in these animals. Rick published a really nice review a couple years ago showing that in fact there's a whole variety of potential diagnostic approaches and predictions of therapeutic responses in immune thrombocytopenia. And these certainly involve T cells, they involve platelet sialic acids, platelet apoptosis, monocyte markers. So we know there's many therapeutic options in ITP, and these therapeutic responses are really challenging to predict who's going to respond and who's not going to respond. And it may very well be that on the left here, it's these cell types within our immune system that may predict whether or not a patient is going to respond or not. And I think there's a lot of work that's now being done on and focusing on these cell types to try and understand predictive responses in patients with ITP. A little bit now about refractoriness. You know, pharmacologists, I have a department, I'm also appointed to the Department of Pharmacology at the University of Toronto. Pharmacologists have been looking at drug refractoriness for 100 years, and it's quite simple to them. Patients, doctors that treat ITP, they can't define refractoriness. I don't know why, but pharmacologists can. And in fact, what it means is you don't respond to a drug. That's the simple definition of refractoriness. One of the reasons why we don't respond to drugs is because there's definitely multi-drug resistant mechanisms within the immune system and within the physiological system of both humans and animals. And there's a whole variety of systems that can be looked at with regards to why maybe a drug is not efficacious in a particular disease. Efflux pumps block penetration of the drug getting into a cell, target modification, and inactivation of various enzymes that metabolize different drugs are all partially responsible for refractoriness to drugs from a pharmacological point of view. Every single one of these mechanisms haven't been done yet, but they can be tested in the active ITP model, and we can try to understand more about multi-drug resistance in these animals. There are several aspects of refractoriness, again, from a pharmacological approach that have been identified in ITP. P-glycoprotein levels tend to be elevated. P-glycoprotein is a drug efflux pump, and it pumps drugs out of cells to protect the host from the toxic nature of the drug. T cells in refractory ITP certainly are altered in some cases, and there's a variety of alterations that have been looked at. And there's a lot of genetic polymorphisms that tend to be associated with drug resistance in ITP, immune checkpoint inhibitors, cytokines, cytochrome P450, and certainly detoxification enzymes. We don't talk a lot about these things in ITP, but they're talked a lot in pharmacological literature. And in fact, I think that we should be focusing more on these types of approaches to understand refractoriness, and maybe we can predict even biomarkers that might predict a person that will or will not respond to a particular drug. So all of these, again, can be tested in the active mouse model. We haven't done any of them. A lot of people around the world are using this model, but we haven't got to the point where these are being tested. But I'm hoping that within the future we'll start to look at and get some data associated with some of these aspects of refractoriness in ITP. Just to remind everybody, platelets, we think about them as the innocent bystander. They're the things that are destroyed in ITP. Well, the thing is, platelets are immune cells too. They're not just hemostatic fragments. They have a whole variety of immune nature to them. They have a proteasome that they can present antigens to T cells. They have all the toll-like receptors, so they are very good at binding infectious particles. They have a lot of mediators, both pro-inflammatory and inflammatory mediators that they release. And they have the biggest source of CD40 ligand in the blood. And in fact, that allows them to very intimately talk with things like neutrophils and B cells. And so we need to think about that, is that can platelets control the immune response to themselves? And that's something my lab's been very interested in. So the question is, do platelets play a role in refractoriness? And this is the classic relationship that we have between the pro-inflammatory Th1 and Th17 responses to T regulatory responses, which are the suppressive responses. When the platelet counts are low, there's high pro-inflammatory Th1 and Th17 responses, and there's very low T regulatory responses and their associated cytokines. Any medication that raises the platelet counts reverses this relationship. So you have now a rescuing of the Tregs and the TGF-beta, their associated cytokines, and you have a lowering of these pro-inflammatory. This is based on over 200 papers now since it was first described about 10 years ago. Just to let you know, T regulatory cells, if you didn't know, this year's 2025 Nobel Prize has been awarded to Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi for their discovery of Tregs. Of course, Brunkow and Ramsdell really looked at FOXP3, whereas Sakaguchi actually described the cell itself. And that was about 30 years ago that they did this. Just for the industry people in the room, just to make you feel like you're really involved, and you should be, is that both Brunkow and Ramsdell are industrial scientists. They started in academics, but they moved to industry, and they discovered the FOXP3 in Ramsdell's company. So certainly industry plays a real important role in understanding the immune system, and a Nobel Prize has been given to industry partners. Platelets also express all of the molecules that we use to target through therapies. They have the MPL receptor, which is the thrombopoietin receptor. They have SYK, they have BTK, they have mTOR. They have a whole variety of these molecules. And so the question then becomes, can platelets play as an immunological sink? When you give the therapy, the platelets soak it up because they express the molecules that the therapy targets, and do they inactivate the particular therapeutic responses? And that's one of the things that we're trying to understand in the animal model. So in conclusion, there's several animal models of ITP. All of them are very valid and robust. The active ITP model, I think, is superior in that both pathogenic mechanisms related to T cells and B cells can be examined. And all therapies in ITP can be tested in this animal model quite readily. The active ITP model was developed to mimic several aspects of the human disease. They have Treg defects, cytokine production defects, and they respond to treatments very similarly to how humans respond. So I think that this model really can be used to investigate critical questions involving refractoriness to ITP. And I'd be willing to discuss this with you. I think that this is the one approach that we can do to try and understand the pathophysiologic mechanisms of refractoriness in this disease. So I just want to acknowledge the people in my lab that were responsible for some of the data production, my collaborators around the world, and I really appreciate your time, and I'll be glad to talk and ask any questions or answer any questions. Thanks very much.
I'm glad to see you upgraded some of your graphics, by the way. Thank you.
Better, eh?
Thank God for that. At long last. Well done. You and then Rick and then Waleed. So we just need to watch time. We've a lot of talks to get through in this day, so we don't want to slip too much.
Yeah, very nice talk, John, as usual. I was wondering about the what you said about the polymorphisms and drug resistance. Of course, we have done in the past a lot of studies on Fc gamma receptor polymorphisms and ITP, but I'm not aware of actually that has been studied in the context of drug resistance.
So there are studies that have looked at, for example, P-glycoprotein, the genetics of in terms of deficiencies and changes in RNAs associated with P-glycoprotein. So it's just starting, and I think that that's what we're going to have to look at in terms of the phenotype of these non-responsive individuals.
Yeah, I think that's actually quite relatively straightforward to do.
Yeah, it is. Waleed.
Great talk as usual, John. What does it take to test a drug in a mouse model once
Money.
one of these? Why money? I mean, they're everywhere. Okay. So if we want to test anti-CD38, because really, I think we can find a biomarker there.
Yeah. No, I agree. I mean, the thing is that it hasn't been done yet, CD38. But, and I'm not quite sure whether daratumumab, if mice, I think the CD38 is fairly monomorphic between mice and humans. So I think probably we'd be able to use daratumumab. I don't know, Rick, have you used it at all?
It has been attempted to another group to do it.
Yeah.
So it's the next one on the list in terms of, trying to understand.
But yeah, I'm going to show some data from the Chinese CM313, but they look at certain things. Probably we can look at other things.
Yeah.
Well,
Thomas.
very, very interesting talk as always. I think that I have much more than a question. I have several commentaries, I must say.
Oh no.
Oh no, yeah. I think that we are quite far from the discovery of anything, because we are looking for predictors of a patient to be diagnosed of ITP, as Lamia published last year. So I think there are not very important publications, but the one from Rick reflecting the possibilities of leading directed therapy, empirical therapy, instead of our empirical therapy. Well, the moral here is that we are not able to know which patient is able to respond to a drug or not.
That's right.
I think that, for example, I get notice of your slide with the drug resistance polymorphisms and so on. You know that last year we published our data on blood PTH with polymorphisms in almost 20 or 30 downstream signaling mechanisms of action. So the question is, what is the main target we have to try to discover, to look into, in order to discover, for example, which patients are going to respond to steroids or not.
Yeah.
Something similar, so simple as that.
I mean, you're right, and it's a very good question. The problem, again, comes down to those first slides that I showed, is that you don't know if your patient's truly primary or not. I mean, because they may have a secondary disorder that's changing the whole pathophysiology of the disorder. They've got thrombocytopenia, but they have something else, and that might really affect how they respond to the therapy. So once we figure out how we can clean this disorder up in terms of, okay, this patient's primary, this patient's secondary, then I think we can really address questions like you published last year and try to understand this. I think that's the critical aspect that most of the basic science labs in ITP are trying to figure out. How do we distinguish primary from secondary? We can't right now.
Can I ask a question, John? You know, one of the problems with ITP, as you know, in humans is the heterogeneity. Oh. Like, how many forms of ITP are there? Is it 5? Is it 50? 500? Have you got heterogeneity in the most population? Are you looking, or is it a clean population, all with the same pathophysiology, or is it going to be multiple forms?
No, it's fairly clean because, again, these are inbred animals. Of course, it's contrived. I'm not going to say that these absolutely are human ITP, but they mimic it in so many different ways. But it is a clean system.
But isn't that slightly disadvantageous in the sense that you want to have a mixture?
Yeah.
You may want some B cell types or T cell types or NK.
So I would argue that, in fact, the model is probably predicting primary ITP responses because it's so homogeneous in terms of how it responds.
And is it predominantly B cell in this, or is it? Again,
both. I think T cells play just as important role as B cells
within the same mouse.
Yes, yeah. In every mouse that we've looked at, whenever there's an antibody in that mouse, there's a T cell, and that's true in humans too. Even though it's more difficult to think, these antibodies are all IgG. If you remember your basic immunology, IgG molecules are only produced by T cells telling the B cell what to do. Mice that have no thymus, athymic nude mice, patients with DiGeorge syndrome, they can't make IgGs because they don't have T cells. So it's, to me, a no-brainer that if there's an antibody response there's a T cell response too.
But I'm just thinking, we often say that if a patient has a T cell type ITP, you might give out a T cell type therapy rather than an anti-B cell. Yeah. It's just we haven't got that level of sophistication that maybe you have in the mice.
No, we don't in humans, that's for sure.
What is the connection? This is an easy question, I say. What is the connection between Tregs, Bregs, and, for example, antibodies? We know that not a minority of patients who suffer ITP have antibodies, and probably the combinations with Tregs, down regulation of Tregs.
Well, that's the connection. Yeah. That is the connection, in that it seems that in patients with active disease, there's a faulty Treg and Breg pattern in these patients.
But if I understood well, probably not in all ITP patients, the Tregs play the key role there in the pathophysiology of the disease.
Nothing is 100%, Thomas. Nothing. Except death, of course.
I could accept.
And taxes, yes. And taxes.
I could accept 80%, for example.
Well, I think those are hopefully reasonable numbers that we can approach eventually when we figure out or maybe discover a biomarker that we can distinguish primary from secondary, for example. And I think if we could do that, my bet would be that in a high percentage of those primary patients, there's a defect in the Tregs that allow for the autoimmune response to become activated.
But up to date, the key role in the pathophysiology of ITP remains to be Tregs, right?
Well, I think they play a big role in the pathophysiology. You won't get
because all the downstream signaling may play a role. But
it's the suppressive aspect of the Tregs in their role, and because they're not working properly, it allows for autoreactive immune clones to become activated, and that's the problem with all autoimmune diseases: type 1 diabetes, multiple sclerosis. They've all been shown to have Treg defects, and this is why the Nobel Prize was given to these cells, because they're so important in suppressing our autoimmune responses.
I think we'll stop there. John, thank you so much. That was a very interesting presentation.